Good morning, everyone. I'm Dr. Ana Karen Carrasco Laserna of the DLSU Central Instrumentation Facility at the Laguna campus. So I'm an academic service faculty and equipment specialist for the chromatography laboratory. And today I'll be talking about chromatography and mass spectrometry for sustainability research. So first, we talk about chromatography. So from the word chromatography, chroma, and graph. So it's here, as you can see, you see a separation of different colors. So this is actually a sample of a plant pigment or dye, and through the use of a solid phase, like here in a paper and some solvents, you will be able to separate out different components of your plant pigment or dye. And this pretty much sums up the name. So chromatography. So color and graph, a visualization of the different colors or components of a sample. So the use of chromatography actually dates back to the late 1800s to the 1900s, and there had been several people that has been attributed to have first used chromatography. But it is generally attributed to a Russian Italian botanist named Mikhail Svet, uh, who was the first one to make use of column chromatography. So he made use of calcium carbonate to separate out plant pigments, and he was also able to. He was the one who first coined the term chromatography and correctly interpreted the chromatographic process. So, chromatography is actually an analytical technique wherein components of a mixture are separated based on their partitioning or interactions between two mutually immiscible phases. So, these immiscible phases consist of your stationary phase, which is the stationary, it is fixed in place either in a column or in a planar surface, and a mobile phase that moves over or through the stationary phase, carrying the analytes. So this technique is actually used in different laboratories worldwide for purposes of quantitation, identification, and purification. So you'll be able to purify your the components of your mixture using chromatography. So there are different separation mechanisms involved in chromatography. So it can be adsorption. So that means your solute absorbs on the surface of your stationary phase. It can be partition chromatography, wherein your solute partitions between the liquid phase that is that is bonded to the surface of a column or stationary phase. It can also be molecular exclusion, wherein your different molecules are separated based on their size and their ability to go through the pores of your stationary phase. You also have ion exchange chromatography, wherein you have, for example, if you have positively charged stationary phase or resin, you'll be able to retain negatively charged ions or anions. And this is in exchange of some weak ions that is initially attached to the resin. And so that's why it's called ion exchange. You also have affinity mechanism or affinity chromatography, wherein you make use of interactions, very selective interactions between different molecules. So, for example, antigen antibody, um, enzymes, and certain other ligands or molecules, and so forth. Now, you can actually classify chromatographic techniques using different criteria. But here, I will just show you like uh, the overall picture of the different mechanisms that is usually used for the classification. So first is based on mechanism. So as we have discussed earlier, you can classify them based on either they make use of partition, adsorption, ion exchange, or molecular exclusion mechanism. You can, or also affinity mechanism. You can also classify based on the shape of the chromatographic bed, whether it's planar or columnar. And you can classify them based on the physical state of the mobile phase. So this can be your gas chromatography, liquid chromatography, and your supercritical fluid chromatography. And we will focus more on these two here, gas chromatography and liquid chromatography, as these are the more commonly used classification as well as techniques. So first, we talk about liquid chromatography, particularly high performance liquid chromatography. So previously, columnar chromatography involves a stationary phase that is in a column, and then you separate out the components of your sample as the solvent is passed through the column. Now, this movement or flow of the solvent through the column is mainly through gravity, and sometimes it takes quite long to get the separation done. And so subsequently, they employed pumps to push the liquid or the solvent through the system. And this is where HPLC was developed. So this makes use of small particle columns with which the mobile phase is pumped through at high pressure. So this is more of a homemade or like an in-lab developed chromatographic HPLC system. Uh, Now we have more commercialized and advanced HPLC systems. So these are examples of newer HPLC systems, and you also have now 2D or two-dimensional LC systems, wherein you can make use of two different types of two columns of the same or different types to further enhance the separation of your components. So in terms of the components of the chromatography system, you have your solvent, the mobile phase that is placed in a reservoir. This is pushed through the system by the pump, which serves as your solvent manager and solvent delivery system. If there is a need to mix different compositions of your mobile phases, then this will be the one in charge of like mixing and making sure that you have the correct composition of your mobile phase at any given point of time. Then you have your autosampler, which introduces your sample into the chromatographic system, the HPLC column where the chromatographic separation happens. Then after they are separated, they get detected through the detector. These can be of different types of detector. And an electrical signal is transformed to chromatographic peaks, as can be seen in this computer data station. In some cases, whatever has been passed through the HPLC system goes to waste. In other cases, wherein, for example, if you make use of a mass spectrometer detector, then that system is destructive in the sense that whatever passes through the chromatography system goes through the mass spec and it gets evaporated and ionized. So you don't get any waste or retrieve your samples anymore. Now, in terms of the mechanisms employed in HPLC, these are the primary modes. So you have normal phase, which allows you to separate polar analytes. So you have a polar stationary phase and a non-polar mobile phase. You also have reverse phase LC, which involves the partitioning of your analytes between a polar mobile phase and a non-polar or hydrophobic stationary phase that is bonded on a solid support. So previously, your solid support, in your solid support, you have a coating of your liquid stationary phase, but now they have already made use of covalently bonded hydrophobic groups for the stationary phase. So here in reverse phase, the nonpolar analytes gets retained more. You also have ion exchange chromatography in HPLC, wherein you have an exchange of analytes with the counter ions of the ionic groups attached to a solid support. So in this case, you have sulfonic functional groups that are attached to your stationary phase or silica support. This attracts your cations to replace your sodium ions. And you also have size exclusion chromatography or gel permeation chromatography. And this is particularly useful for the analysis of polymers or mixtures of polymers or biopolymers. This causes exclusion of your bigger molecules uh, while your smaller molecules permeate or goes through the pores. And so you will see from the separation, those that are big gets eluted out first, comes out first, whereas those that are smaller and passes through the pores will come out later in your chromatographic separation. There are other chromatographic techniques that can be employed, such as affinity chromatography, chiral chromatography, hydrophilic interaction chromatography, hydrophobic interaction chromatography, electrochromatography, as well as supercritical fluid chromatography. And these will depend on what is your analytes of interest and what will be the most suitable chromatographic mechanism for them. Now we also have what is known as preparative HPLC. So what is the difference between preparative and analytical HPLC? So when we talk about preparative HPLC, the distinction previously was based on the column employed, because previously for preparative HPLC, you have to make use of very large columns. And then, so if it's preparative, employs very large columns. If it's analytical, it's like the smaller columns. But of course, this is not proper because even preparative LC can have like small columns as well. And so they defined it better. So now, of course, analytical HPLC is used for the separation of components for purposes of detection only. Whereas your preparative HPLC allows you to separate components for detection as well as for subsequent collection. So after you collect your different components, different separated components, you can use it for further purification, recycling through the preparative HPLC system, or for further isolation and characterization. And so this is very useful if you want to isolate certain components of your sample, such as in natural products research, wherein you want to isolate active components of the natural products, and so forth. These are the different chromatographic modes employed for the different types of molecules. As you can see, there is a prevalence of reverse phase chromatography for any type of molecules, whether it's macromolecules or organic small molecules, and even preparative. So it's mainly a reverse phase as well as normal phase. For macromolecules, again, gel permeation chromatography is commonly employed. Now, in chromatography, so again, when you have mixtures, right, especially in complex mixtures, you want to separate out the different components as much as possible. And so this can actually be achieved using different parameters in chromatography, but mainly through one of them mainly is through the use of small particle size columns. And because with smaller particle size columns, you'll be able to increase your theoretical plate, and with higher theoretical plate in chromatography, this actually allows you to resolve your different components more. So as you can see from here, if you have bigger particle size in your column, then the separation is not so good. But if you have smaller particle sizes, particles in your column, then you'll be able to have better separation and narrower peaks for your analytes. Unfortunately, with this, there is also a drawback, because with smaller particle size, then you get higher back pressures. And previously, chromatographic systems or HPLC systems cannot withstand the high pressure that is brought about by the use of these smaller particle size columns. And so they resorted to, subsequently, they have developed what is known as the UHPLC. So UHPLC is known as ultra high pressure liquid chromatography. This is one of the most important developments in modern HPLC, and this has been pioneered by James Jorgensen in 1997, and later on by Milton Lee. The first commercialized UHPLC system was named UPLC, or Ultra Performance Liquid Chromatography, by Waters. And this UHPLC actually provides you faster and better resolution separations, and also help increase your sensitivity because you get narrower peaks. So in this case, you will be able to get higher intensity signals. So this enables you to make use of very small particle size columns, such as 1.7 or 1.8 micron particle size columns. So Typically, in terms of the back pressure, as I mentioned earlier, the usual HPLC columns can only stand up to 400 bar. Using UHPLC systems, you'll be able to make use of columns that and have pressure limits of 600 to 800 for intermediate UHPLC, or 1,000 to 1,500 for UHPLC. Now, different detectors can be employed for HPLC. So chromophore containing analytes, we usually make use of UVVs or diode array detector. The detector is set at the maximum absorbance wavelength, and of course, it's best to make sure that your solvent that is employed does not absorb at the wavelength of that you're going to use. And so it's best to select wavelengths that are greater than 220 nanometers. So for diode array detector, you make use of the different wavelengths of the UVVs region. You'll be able to get a three-dimensional chromatogram such as this. If your analytes are non-chromophoric, you have to either derivatize your analytes, which can be tedious, or make use of other forms of detectors such as refractive index, evaporative light scattering detector, charge aerosol, fluorescence, and other is your option of mass spectrometer. But this is for ionizable species. So these are just some of the mentioned detector together with their mass detection limits. So as you can see, you'll be able to obtain very low detection limits with the mass spectrometer. Um, you have RID, which is relatively more universal, and the others will be of higher detection limits. Now, in terms of the instruments that we have at CIF, we have an analytical HPLC or NGPC, or gel permeation chromatography system with multiple detector. So we can make use of UV variable wavelength detector. So the wavelength is from 190 to 600 nanometers. And then we also have refractive index detector, very useful for sugars, which doesn't have a chromophore and it's not very ionizable in mass spec. And then for polymer analysis, we have light scattering and viscometry detectors that can allow you to determine molecular weights based on their size, and also provide information on their shapes as well as behavior. We also have a semi-preparative HPLC with UV variable wavelength detector. So these can allow you to, again, fractionate different components of your mixture. So for further characterization or for further purification. So very useful for natural products research. Lastly, we have our UHPLC system that is coupled with a quadruple time of flight mass spectrometer. Again, we will talk more about this later, but again, this will allow you to employ columns that are of smaller particle sizes for better resolution or chromatographic separation. Okay, now we go to gas chromatography. So in gas chromatography, the components of a vaporized samples are separated based on the differential distribution between a gaseous mobile phase and a liquid or solid stationary phase that is held in a column. In contrast to the other chromatographic techniques wherein you have interactions between the analytes and your mobile phases, in gas chromatography, the mobile phase does not interact. So the mobile phase has to be inert. So these are the two types of gas chromatography. You have gas solid chromatography and gas liquid chromatography. In gas solid chromatography, your stationary phase is solid and the mechanism employed is absorption. This is useful for low molecular mass gases like hydrogen sulfide, carbon monoxide, nitrogen oxides. Gas liquid chromatography, you have a liquid immobilized on a stationary phase for your stationary phase. Liquid immobilized on a stationary support for your stationary phase, and this employs partitioning. Now, this is very useful for the separation of your volatile organic compounds. So a main parameter for gas chromatography is that the analytes must be volatile and thermally stable. And in terms of like naturally volatile and thermally stable analytes, this will only comprise 20% of your known compounds. But of course, you can also perform derivatization. And this will allow you to make your analytes volatile and improve the chromatographic separation as well as detection. These are the components of your GC system. So you have your gas cylinder containing your inert carrier gas. You have a regulator to regulate the flow of your carrier gas. The traps to filter moisture or other contaminants from your gas. And this goes through your chromatographic system, your GC. Your sample is introduced through the autosampler. Then chromatographic separation occurs in your column oven, within your column oven in the column. And then you have a detector for the detection of the components. And you have, again, a computer data system to record the separation, that acquire the data from your chromatographic separation. So you have different modes of GC separation. So you can be, again, adsorption or partitioning. If you have gas solid, then it's adsorption. Gas liquid will be partitioning. Now, there are different means to introduce your sample to the gas chromatographic system. It can be direct liquid injection, but this is, of course, limited to volatile solvents and volatile components. Um, you can make use of headspace sampling, wherein you sample the volatile components that are present in the headspace of your vial. So your needle doesn't touch your solution. Rather, it will collect sample from the headspace of your vial. So dynamic, there will be inert gas flowing through while you're before you sample. Purge and trap, on the other hand, is that the inert gas passes through the solution in order to release your volatile organic compounds into the headspace. And then subsequently, there is an adsorbent or cold trap to concentrate your analytes before they are sent off to the GC. You also have thermal desorption, which is useful if you have sorbent collected volatile or organic compounds. So afterwards, they can be desorbed from the sorbents and subjected to GC analysis. There is also pyrolysis. So you just put your sample within this um, pyrolysis furnace, and whatever has been degraded from the pyrolysis can be subjected for analysis. In carrier gas that is employed for GC, most commonly helium is used. Hydrogen can provide better separation, but it's highly flammable. So there is a consideration. This is a consideration that you have to take note. So, but most commonly it is helium. There are different types of columns that is employed for gas chromatography depending on the analytes of interest. You also need to take note of the maximum temperature that the column can withstand. In terms of like internal diameter, so we have capillary columns of like very small internal diameter such as 0.1, 0.25, and this provides you with better resolution and speed. In terms of column length, so longer, then it's better. But of course, your chromatographic separation or chromatographic run will be longer, and so you have to uh, have some compromise on that. Different detectors can be employed. You have the universal detector for carbon, such as flame ionization detector. You have those that are more selective in terms of like the presence of certain atoms, like NPD for nitrogen phosphorus detector. Um, for phosphorus or sulfur, you have flame photometric detector and so forth. Of course, you can also make use of mass spectrometer. In mass spectrometry, so this one is first devised by Sir J.J. Thomson as he tried to separate particles of different masses within a beam and determine their relative quantities. So it is important. It is an important technique to weigh ions in gas phase, whether it be individual atoms or molecules. So basic principle involves the motion of charged particles in an electric or magnetic field. Separation is based on the mass to charge ratios. Applications include atomic or molecular weight determination, structural characterization, gas phase reactivity studies, as well as the qualitative and quantitative analysis of components in the mixture. So instrumentation involves the introduction of the samples into the MS system, either by direct insertion of the samples into the MS or through um, flow injection or liquid chromatography or capillary electrophoresis. Subsequently, it goes through the ion source to produce the ions before it goes into the vacuum component of the MS, wherein you have the mass analyzer to separate your ions based on mass to charge ratio via magnetic or electric field. Then you have a detector that counts the ions and produces the signal of the separated ions. So subsequently, you have an instrument control and data system to control the instrument and process the data and produce the mass spectra. Now, the vacuum system is important to minimize ion molecule interactions, scatterings, and neutralization of the ions. So again, sample introduction can be direct insertion of the sample into the vacuum system or through infusion, such as when you use chromatography. There are different types of ionization techniques employed. For GC, it's mainly electron ionization and chemical ionization. Electron ionization allows you to have very reproducible fragmentation patterns that can be used for the identification of your compounds based on available spectral libraries. For LCMS, it's mainly electron spray ionization that's employed that can be used for more diverse range of analytes from neutral to ionic, from relatively nonpolar to polar, and up to 10 raised to 5 molecular weight molecules. So some important terms in MS is high resolution, is mass resolution and mass accuracy. So mass resolution refers to the ability to separate adjacent ions. So the higher the resolution, the narrower the peaks, and the more resolved your ions are from each other. When we talk about mass accuracy, we measure the accuracy of the mass information obtained by the MS versus the true exact mass. So this is very useful if you want to identify your molecules or atoms. So you have different types. So different types of mass spectrometers are identified based on the mass analyzers. So after the ions are formed, they are accelerated into the mass analyzers, and they can separate the ions according to the mass to charge ratio. So you can have continuous analyzers like quadrupole filters and magnetic sectors that wherein the ions will just pass through the mass analyzer and they are separated or filtered. Whereas you also have pulsed analyzer wherein ions are released into the mass analyzer at specific times. So these are examples of mass spectrometers that are named based on their mass analyzer. So this is a triple quadrupole. So you have, and you also have your quadruple time of flight and your orbit trap. So you can also do tandem MS wherein you can fragment your ions first, filter your ions, subject it to fragmentation, and then filter the ions that will be detected. So this is more for purposes of highly selective and highly specific detection, wherein you single out your analyte of interest, fragment it, then you can pick fragment ion of interest for a very specific detection. So this is a comparison of different mass analyzers employed. So you have like low resolution, low mass accuracy, but they can provide configurations that will allow higher sensitivity detection. You also have high resolution systems with very high resolving powers. And they can also provide you with tandem MS settings for high sensitivity detection. So we have different MS acquisition modes. Again, this will you have those that are more of a scanning. So whatever ions of a particular mass range is defined, you will be able to detect them. You have more targeted analysis, wherein you make use of fragmentation to isolate ions of interest. And you also have selected ion monitoring in case of single mass analyzers. So in atomic analysis or elemental analysis, earlier Chris has talked about um, inductively coupled plasma, OES, or optical emission spectroscopy. So given that you also generate gaseous ions in ICP, these are actually very useful and can be detected using mass spectrometry. So in terms of detection limits, as you can see, ICP-MS can provide very low detection limits for your elemental analysis. And this allows like simultaneous analysis of your different ions. This is just a schematics of an ICP-MS instrument. So you have the ICP in front for the generation of your ions. Then the back part, it will be your usual mass spectrometer, such as single quadruple, triple quadruple mass spectrometer. So we have a Shimadzu inductively coupled plasma mass spectrometer. This has a quadruple MS. It can be used for simultaneous analysis of multiple elements. It can be used for analysis of environmental, drinking water, wastewater, for analysis of heavy metals. And this again covers different aspects of the UN SDGs for good health and well-being, clean water and sanitation, life on land, life below water. Now, chromatography can be coupled with MS, as we have seen earlier. They can provide higher sensitivity and specificity of detection. This provides more definitive information on mass and structure. In terms of components, basically it's the same. You just have the HPLC or GC in front. It provides additional dimension of components resolution. So just like in PDA, wherein you get a three-dimensional plot, you will also be able to obtain a three-dimensional chromatogram or spectra from your uh, coupling of chromatography with MS. And this information can actually be used for the identification of your compounds. So you can do untargeted analysis, wherein it's more of the exploratory analysis, and this is usually performed with high resolution MS, or you can analyze target analytes of known compounds or class of compounds, and this makes use of tandem mass spectrometry. So this allows you for higher sensitivity detection. So in CIF, we have UHPLC QTOF as well as GC that is coupled with the mass selective detector, which is basically a quadruple MS with headspace sampling and liquid injection. So in terms of applications of chromatography and mass spectrometry in sustainability research, there are a lot. So from here, you can see is an example of the use of GCMS and HPLCMS for the detection of different pollutants in the Danube River. And these actually addresses sustainable development goals, sustainability development goals such as in good health and well-being, clean water and sanitation, life below water, and life on land. You can detect certain pollutants such as triazoles, pharmaceuticals, pesticides, flame retardants, etc. You also can use it to detect drugs or drug substances of abuse and their analogs in targeted detection, such as in this case, they were able to detect these fentanyl analogs in hair, and this can address your UN SDG 16. And actually, in cases wherein you're not, because currently there are a lot of developments of analogs or derivatives of these drugs of abuse, even in order to elude detection, untargeted analysis can allow you to identify those. In food and nutrition research, there are a lot of applications of mass spectrometry as well, and these address UN SDGs 2 and 3 in terms of zero hunger and good health and well-being. Food safety, there are also a lot of applications in terms of detecting pesticides and herbicides in fruits and vegetables, antibiotics and veterinary drugs in meat products, effect of different processing methods in food products, the detection of chemical leachates from food packaging materials, detection of adulterants in food supplements. Mass spectrometry-based techniques can also be used for omics application. So this addresses several UN SDGs as well, such as zero hunger, good health, clean water and sanitation, industry. innovation in infrastructure, in sustainable cities and communities, life below water, life on land, because these omics technologies are actually useful in studying different living organisms, and so it is actually very applicable in different areas of research and in sustainability, in addressing sustainability goals. Now, this is an example of a study that we did in urban agriculture to determine the effects of biochar for in the growth of these vegetables bok choy. So again, these can address several UN SDGs, responsible consumption and production, because this involves the use of biochar, which is coming from agricultural and municipal wastes, urban farming, and zero hunger, because this involves the production of your vegetables in urban farming settings. And we used LCMS LCMS and NMR to determine the effects of the use of biochar to the bok choy. And so here we find that actually there is an increase in levels of carbohydrates, decrease in amino acids, but in the LCMS results, we find that there is an increase in productions of glucosinolates and flavonoids with the use of biochar. Currently, in the situation now, MS can also be used in the study of SARS-CoV-2 virus and COVID-19 research. It is very much the different omics technologies are um, currently being studied in terms of like patients that are infected, asymptomatic, under recovery, to see like what are the effects of um, having this COVID-19 disease to the person. And there are also studies involving cell culture, and mass spectrometry is very much used in these types of studies. So in summary, I've provided an overview of chromatography and mass spectrometry. Different chromatography and mass spectrometry configurations are available depending on your analytical requirements. You can couple chromatography and mass spectrometry, and this provides powerful analytical instrumentation for a diverse range of applications. Chromatography and MS instrumentation can provide or help address um, several of our UN Sustainable Development Goals. So depending na lang po in terms of your particular research, we can see how we can make use of chromatography and mass spectrometry to address your research objectives. So if this is the end of my presentations, if you have any questions regarding the use of chromatography, please do email us at chromatography.lab@dlsu.edu.ph. And you can also email me in my work email, anna.laserna@dlsu.edu.ph. So that's all. Thank you.